so now the next part of the paper's methods was on sequencing the COVID virus, and then we'll show you the images later and the results and so on. So far, none of the steps covered even remotely show any common sense demonstrations right here of COVID isolation, let alone what the paper is trying to achieve. Let's now take a look at the next section of the paper, which is on sequencing the COVID virus from the cell cultures. So note that I will not go over too many of the definitions of the specific industry methods for our DNA sequencing mentioned in the paper, because again, I'm more focused on the physical isolation and so on. Alright, so RNA extracted from bronchoalveolar lavage fluid and culture supernatants was used as a template to clone and sequence the genome. And we used a combination of Illumina sequencing and Nanopore sequencing to characterize the virus genome. And yeah, basically, so it takes the RNA from the airway fluid samples and the cultural supernatants, and then somehow assumes it's a template, or it's used as a template, and then it clones a million times, and then somehow you sequence the genome using these two technologies. And Illumina sequencing involves generating millions of short sequence reads, about 150 to 300 base pairs in length, all in parallel, so doing it all at once. And Nanopore sequencing is a new technology that can generate very long continuous sequence reads, several thousand bases in length. And the short reads in Illumina sequencing helped correct errors in the long continuous reads in Nanopore sequencing. Yeah, so use these in conjunction. This one is short reads, this one's long ones but has errors. And then you could correct them using these shorter reads. So continuing further in the paper, so sequence reads were assembled into Contig maps, which are a set of overlapping DNA segments, with the use of CLC genomic software version 4.6.1 and then CLC bio. And note that a contig, from the word contiguous, is a continuous sequence of DNA that has been assembled from a set of overlapping DNA fragments called reads. And the word contiguous means objects are physically close together, as opposed to continuous means a smooth transition and so on. And a contig map is a graphical representation of the physical organization of contigs in a genome or chromosome. For example, here's a good video tutorial. Here's for example, if you have this chromosome right there, then if you get all the sequences in it, you're going to have a lot of regions overlapping when you're trying to obtain each of these shorter reads. And then when you form a continuous one, that's the contig, which you would have these overlapping ones. So you would have this overlapping over here and so on. I'll just change this color to red. So you have this overlapping, and then you would form a single continuous one. Yeah, like that. And then just get back to the black pen. All right, so we have that. And continuing further, so yeah, video tutorial contigs are continuous sequences formed from overlapping sequences. All right, so continuing further in the paper, so specific primers were subsequently designed for PCR, and 5' or 3' race or rapid amplification of cDNA or complementary DNA ends was used to fill genome gaps from conventional Sanger sequencing, and these PCR products were purified from gels and sequenced with a big dye Terminator V 3.1 cycle sequencing kit and a 3130XL genetic analyzer in accordance with the manufacturer's instructions. Interesting. Let's just break this down here. So specific primers were subsequently designed for PCR. So a primer, again, as I went over it earlier, is a short single-stranded nucleic acid used by all living organisms in the initiation of DNA synthesis. And the 5' or 3' right here, this race right here, refer to orientation of a single strand of a nucleic acid, and have this over here. I just showed this illustration. So a furanose or sugar ring molecule with carbon atoms labeled using standard notation. The 5' is upstream, so you go from here 1, 2, 3, 4, 5, and the 3 is downstream. And DNA and RNA are synthesized in the 3, in the 3 to 1 in the 5 to 3 direction. So that's how they're synthesized, starting from here, going downwards, like that. So hence, it's important to know the orientation. And going further here, so complementary DNA or cDNA is DNA synthesized from a single-stranded RNA template by the RT or reverse transcriptase enzyme. In Sanger sequencing or chain termination sequencing involves replicating DNA using modified nucleotides or dd-NDPs, to terminate the DNA, cDNA, in this case, chain elongation, and allows for generating a series of DNA fragments that differ in length by a single nucleotide. Interesting. So basically, you would replicate, but one sequence, one nucleotide long at a time, and by these special ones that terminate after you do that one, which is quite interesting. And also, so this primer part right here, here's another illustration of primers. So schematic, the DNA replication fork. So you have RNA primers labeled at the top. So here's RNA primer. And there's a video animation of showing how this works. And the fork splits the DNA double strand. So this fork splits these into two, like that, double strands. And then the leading strand replicates continuously. So then this replicates continuously. And then you're going to replicate this, matches up with this. And the leading strand replicates continuously, while the lagging strand replicates discontinuously. This one here adds one at a time, like that, or it puts this primer like this. Yeah, so this is this is lagging one, this is the leading strand, and this one adds a primer, and then synthesize adds a primer. I believe that's how it works. But anyways, this one is more discontinuous. This one is continuous setup. All right, let's continue further. So continuing further in the paper, multiple sequence alignment for the 2019 nCoV and reference sequences was performed with the use of muscle. Phylogenetic analysis of the complete genomes was performed with RAXML, and then bracket 13, with 1,000 Bootstrap replicates and a general time reversible model used as a nucleotide substitution model. All right, let's make sense of all this. So multiple sequence alignment is a process of aligning the genomic sequences to identify similarities or differences. And then this phylogenetic analysis or just phylogenetics, is the study of the evolutionary history and relationships among or within groups of organisms. And then Bootstrap replicates are a statistical technique to estimate the probability of a phylogenetic tree by generating multiple random samples of the original data set. And substitution model is a model describing the changes in DNA sequences over, quote, evolutionary time. And a time reversible model does not care which sequence is the ancestor and which is the descendant. Anyways, that's just a phylogenetic analysis of these genomes and so on. And let's go over a summary of sequencing the, quote, virus. So here's a quick summary of the, quote, virus sequencing procedure in this paper. So RNA from patient airway fluid plus cell culture supernatants were extracted and assumed to be from a, quote, virus. The genome of this RNA assumed, quote, virus was produced using a combination of Illumina sequencing, which produces millions of short sequences, and Nanopore sequences, which produces continuous long sequences. And the short reads are used to correct errors in the long reads. And primers, or short nucleic acids that initiate DNA synthesis, were designed for PCR tests. And then DNA fragments from PCR were then sequenced. And then going further, the, quote, virus sequences were compared with reference sequences, and lastly, statistical analysis was performed on the likely, quote, evolutionary history of the, quote, virus genome. And now the question still remains: where is this, quote, virus? How do they know there is a, quote, virus? Why is the paper throwing the word, quote, virus around so casually? #questions. And now the next